Hello everyone, in this lecture we are going to study purification of viruses. It is necessary to purify viruses to study virus structure, virus reproduction, etc. A number of techniques are available for purification of viruses. Due to the virus features such as larger size than proteins, more stable than cell components and presence of surface proteins, techniques used for virus purification are similar to the techniques used for isolation and purification of proteins and cell organelles. The common techniques are density gradient centrifugation and precipitation. Density gradient centrifugation. This method involves sedimentation of particles in density gradient medium. This method separates particles based on their buoyant density. This method gives much better separation than differential centrifugation. It allows separation of subcellular particles, macromolecules, viruses, etc. and measurement of sedimentation coefficients. There are two methods, namely red zonal centrifugation and isopycnic or equilibrium gradient centrifugation. Red zonal centrifugation. In this method, sample is centrifuged in a preformed gradient. A density gradient is prepared in a centrifuge tube before centrifugation. Various substances are used as a gradient material. These includes sucrose, cesium chloride, cesium acetate, glycerol, phycol and ludox. Density gradient is prepared by layering solutions of continuously decreasing density over each other. The highest density solution is at the bottom of centrifuge tube, whereas lowest density solution is at the top of the centrifuge tube. The virus sample to be separated is layered on the top of gradient. Centrifugation is carried out at relatively low speed for a short time. Sample particles travel the steep gradient based on their sedimentation rate and form discrete zones. This method separates particles which differ in size but not in density. The largest viruses move faster down the gradient. The centrifuge run is terminated before any of the zones reaches the bottom of gradient. The zones in this gradient are stable due to absence of diffusion and convection currents. Collecting fractions from the bottom of the tube isolates various zones. Isopycnic or equilibrium density gradient centrifugation. In this method, the density gradient forms during centrifugation. The sample to be separated is dissolved in a solution of cesium chloride or cesium sulfate. This mixture is distributed uniformly in a centrifuge tube. Centrifugation is carried out at high speed for a long time. The cesium salt redistribute under the influence of relative centrifugal force to form a continuously increasing gradient from top to bottom. The sample particles move the gradient at a region where the gradient density is equal to their respective densities and form discrete zones. This method separates particles which differ in density but not in size. Viruses can be separated from other particles only slightly different in density. Precipitation. Like proteins, viruses can be purified through precipitation with concentrated ammonium sulfate or ethanol or polyethylene glycol. Initially, sufficient concentration of ammonium sulfate is added so that virus will precipitate. The precipitates obtained once could be redissolved in some suitable buffer and reprecipitated. In this manner, more and more purified preparations are obtained. The precipitated viruses are collected by centrifugation. Viruses sensitive to ammonium sulfate are purified by precipitation with polyethylene glycol or ethanol. Precipitation can also be achieved by adjusting pH of the suspension. Proteins possess two types of surface charges, positive and negative. Once the charges are neutralized, the proteins will precipitate out. The pH at this point is called as isoelectric point. pH can be adjusted by adding more cations and anions in the form of salt solutions. These ions neutralize negative and positive charges respectively and the isoelectric point is reached. Thus proteins are precipitated out. Some viruses have outer protein coat. They can also be precipitated in this manner. Alpha-alpha mosaic virus is one of the first viruses to be precipitated by this method. Thank you.